everyone, and welcome to the launch of the Fitzroy Partnership for River Health's 11th report card for the water year 2020 to 2021. Thanks everyone for logging on today and for those attending in person. Before we start, just a couple of housekeeping items. For those online, if you do have any issues with the technology, please put some comments in the chat, and Nicole, our communications coordinator, will be monitoring it and we'll try and troubleshoot any issues. We're using Zoom webinar today, so only the speakers will have access to microphones, but we will be doing a Q&A session at the end. So if you do have any questions during the way, please put them into the chat. For those who are here today, the facilities are located through the white door on the right. And if there is an emergency, please follow the staff. The muster point is by the Telstra building towards the right. So please join me in acknowledging the First Nations of the country in which we are meeting today. For those of us in Rockhampton, we're meeting on Durable country. The map that you see on the screen was developed in 2021 with assistance from Hayley Young, FBA's Indigenous Engagement Coordinator. The purpose is really to develop a stylised map of the First Nations in the Fitzroy region for the purpose of providing a first point of orientation for their diverse audiences and partners. As you can see on the map, the main waterways and 17 First Nations groups are depicted. We recognise the diversity of First Nations people within the basin. We acknowledge the important role they play as custodians of the land and water within the Fitzroy Basin. We pay our respects to elders past, present and emerging. The Fitzroy Partnership for River Health is committed to continuing to develop a positive, long-term and mutually beneficial relationship with First Nations elders and peoples within our region. So to start off today, we thought we might take a look back to see where we started and where we're going into the future. So after 11 years of reporting, where we started was in 2008. Following significant flooding within the region, there was community concern surrounding water quality and the ability to obtain, knowledge of monitoring within the region. So a report was commissioned in 2009 to look at how we could improve this. And out of that, the Fitzroy Partnership for River Health was established in 2012. The role of the partnership is to facilitate improved monitoring and to collate and assess data and information to produce a publicly available report on waterway health. The partnership was the first regional report card to be established in Queensland and continues to play a crucial role in educating and informing the community about the health of their waterways. Since our formal establishment in 2012, the partnership has maintained the integrity of the original report card, its data, indicators, and this has allowed us to collect over 10 years worth of water quality data. We're currently reviewing the data set and we'll have some recommendations for our partners in management of waterways following the release of this analysis later in the year. In addition to this, the partnership has built on its solid foundations and evolved with the needs and wants of the community, developing additional useful reporting tools, such as water for drinking, crop and agricultural use, and we'll hear a little bit more about those today. Our citizen science program continues to provide opportunities to engage with the community and to provide regional communities with the knowledge to sample and take care of their own creeks and waterways. You'll also see this year that we've reintroduced reporting on the Inshore marine zone, and we'll hear a little bit more about that as well today. I encourage you to visit our website or get in touch with us to find out more about the work of the partnership. Today is about celebrating our flagship product, which is the release of our 11th report card. It would not be possible without the contributions of all of the partnership organisations who are represented on the slide. In addition to the Ecosystem Health Report Card, the other thing the partnership does so well is bring partners together, and it's important that we share the management actions that are above and beyond legislative requirements. Our partners provide both direct investment as well as waterway health data to support the report card development. They meet regularly and collaborate to improve outcomes for our region. It is important to acknowledge and celebrate our successes. Last year, our 10-year celebration was cancelled due to COVID, but we've definitely not forgotten about that. So we will be celebrating on the 25th of August with a round table and gala dinner. If you don't have an invitation, please contact us so that you can attend. We thank all our partners for their ongoing commitment. After 10 years of reporting, many of our foundational members are still partners, which surely is a testament to the great benefits of being involved. I'd also like to take this opportunity to thank all of our management committee. These dedicated people meet regularly to inform the work of the partnership, and we wouldn't be here without their dedication. The Fitzroy Partnership for River Health really is a true partnership, a collaboration between organisations from government, industry, research, and the community who have all have an interest in the health of waterways within the Fitzroy Basin. A tremendous amount of work goes into compiling the report card every year to provide the Central Queensland community with a complete picture of river health. So in addition to the partners, I'd also like to thank the partnership staff as well as the independent science panel for all the work that they do to pull that report card together. I congratulate the partnership on the release of our 11th report card for the year 2020 to 2021. I'll now hand over to our Executive Officer, Lee, to take us through today's events.
Thanks, Katie. Thanks for the welcome and the introduction. We're really excited today to have this launch of our next report card. So we'll talk about the results in a little moment. Before that, we've got a few other things to discuss. We just wanted to go through some of the highlights since the last launch itself. So what have we been doing since we launched in 2021, our last report card? Have a look at some of the community events we've been doing and so on. We've also got a scholarship that we present each year. So we want to spend a little bit of time today having a look at one of the scholarship winners and some of the research and studies that she's completed. She'll present that to us. And we'll present to the next scholarship recipient who's attending as well today shortly. Then we'll have a look at the actual results. So Eva Abal, Associate Professor, will be online, zooming in to look through the results in a bit more detail. So you'll probably have some questions and things at that stage. Please keep those in mind for our Q&A at the end. We'll look also at the online system and some of the reporting products we have on there. And that should just about cover us, I think, for today. So if I can start with what we've been doing, some of the highlights over the last sort of 12 months or so while we've been out and about in the community. I guess I sort of always, when I think about the partnership, focus on those three kind of pillars that you can see there on the screen. So while the report card that we're focusing on today is our flagship product and the thing that we're most proud of and that we work so hard on, it's the other things as well that we've sort of evolved to become over the last sort of 10, 11 years since we formed. And in particular, I think it's the partners and the organisations that are part of this partnership, I guess. So it's supporting management actions into the future, coming together and collaborating. And it's just wonderful that the partnership itself has those opportunities to provide, you know, a shared platform for discussions. People aren't sitting in silos as much. They're sort of collaborating and discussing ways to improve waterway health. We've also enhanced our community engagement over time too, and Katie mentioned the My Water portal, so I'll have a quick look at that in a moment. But that's the other third sort of pillar of what we do really, is that engaging with community, communicating and educating kids, university students, community groups about what we do, about our transparent independent science in the basin, and the fact that it's trusted information that we're putting out there as well. So having a look at our My Water portal, I've just got some screenshots there from the web page. And I'll have a look in a moment, show you all of the things we've been doing as well. It's quite amazing how busy the team can be, to be honest. I don't know how we get out there when you look at it on paper, but this is the My Water portal. It has a monitoring guide with it that we've produced. We go to schools and we help them to monitor their own creek or their own local waterway. And that has lots of, engaging opportunities. So, everyone used to go and collect tadpoles down the creek when they were kids. It kind of engages everybody from all different ages. And, sure, you can look at our report card, but, what's happening down in my backyard? You can actually get a grade for your backyard and look at that over time and try to put that picture together, I suppose. So we're getting more and more sampling into that as we've developed over years. And people are using that data in schools and things as well to sort of feed into their curriculum, which is wonderful. If you want to get in touch with us and do something like that, please let us know. We've got those resources available. Community engagement, and this is a pretty busy slide, apologies for that, but I had to put it in there. This is the community events we've attended over the last 12 months on behalf of the partnership. Everything from school visits to events, our events ourselves, but we also go to regional events as much as we can, and really trying to go into the outer region as well, not just so much Rockhampton. Probably a couple of highlights are things like the River Festival down on the riverbank here, which has thousands of people attend each day. So that reach is really great, but also presenting at conferences and symposia, getting out and about with the report card results themselves. And also working with our northern friends, the Mackay Sunday report card in St Lawrence. So really developing all of those community events in different ways that we can, and supporting our partners in that as well. Instead of some writing, I've just got the images up now, so you can have a look at some of the team out and about. The Net Free Fishing Zone Forum was a really interesting one as well. So we're kind of building our knowledge in the team about all of that ecosystem health information. We've got our board game still, which is a real hit, and Mindy that some of you might be familiar with, some of you might not be. If you haven't been, you will be soon if you attend the 10-year event, and usually the newer staff member has to make an appearance in Mindy, so that's a bit of a contentious issue at the moment with Braden and Eva, as I might mention later. What next? So that's kind of a bit of a snapshot about our community events. You know about the report card, we'll talk about that today. What are we working on now? A lot, and there's always so much more that we could do. We've just sort of got to focus in on, the most important crucial things, I suppose, that are going to make a difference. Having 10 years of data is amazing. We're learning though that 10 years, that's the sort of ideal in the scientific world as a minimum. It really is a minimum. And we're looking at those patterns of hydrology over time, but we'll be able to really nut down into that 10-year data set and correlate some of the different parameters, which will be really helpful, I think, with our understanding moving forward, particularly with those boom and bust events that we have, in that Fitzroy region. With that, we'll also look back at our program itself, do a bit of a review. It's a good time to do that in the 10-year mark. Is there other things we can do? Let's sit down as a group and discuss how we can better work together for the community and for our partners. Stewardship reporting. So we recently did a social survey of the community. We got quite a few people, involved in that survey around the region, and so we're just going through the results of that too at the moment. So how do people value waterways? How do which waterways are most important to people? What's happening? What kind of management actions do they believe in or not believe in? And hopefully that'll feed some management change as well. And that, I guess, feeds into that human dimension side. So not just the water itself, how good's the conductivity, but how are people in that story? How do people fit into that value story? We've taken over the Drain Buddies program, which has been very active in the region, and FBA and a lot of other groups have set up that Drain Buddy system. It's a little drain that sits in the urban areas in the stormwater and captures the litter. We then take that out. It works as just a science program, take out the litter, and really helps us inform local businesses and local community about what we're finding. Mostly it's cigarette butts, as you'd imagine, but it's interesting to see what things are in different drains around town. So we'll be able to report on that side of it. So moving from the ecosystem to more of the urban space as well within the region. A region-wide monitoring program as well we've been working on, which is really exciting. and of course a new Mindy. So Mindy's been around, that's our mascot, for 10 years. She's getting a little bit stinky inside, as you can imagine, in rocky weather. So new Mindy is on her way, a new improved Mindy, but we just won't tell the kids it's a new Mindy, it'll be the same Mindy. But yeah, so that's what we're looking forward to in the next little while. Move on to the Heart of the Basin Scholarship. And Rachel's just joined us. Welcome, Rachel. So the Heart of the Basin started quite a few years ago. It's in conjunction with CQ University, one of our other partners. Wonderful opportunity for us to support local research and knowledge. To support students, it's designed to be an honours or a RHD, like a postdoctoral or a master's degree support. It's not a big amount, it's $3,000 stipend, but really helps students, if you remember back to those days, to sort of get you through those times. And it allows them to connect with our partners and that wider audience as well. So today we have Natalie Wolf-Rackerman presenting her results from the scholarship that she received last year. Before I hand over to Natalie, who's online, she'll be joining us on Zoom. I just wanted to point out the Heart of the Basin, how cute, clever that is. So Barry Hart was our original chair. He actually wrote the report that was commissioned back in 2008 by the government. And so I think it's really nice how we're still carrying on with Barry's. He couldn't be with us here today, but sends his well wishes. That's an image of him there with two of our past scholarship recipients. So a really great, opportunity for people moving forward with that as well. Natalie, are you online? We might have a bit of a glitchy moment. I'm going to change Natalie's slides for her.
I am here, Lee. Thank you very much.
I'm just going to sit down here like a teacher and change the slides for Natalie as she goes through. You can see her behind me. I'll hand over to you, Natalie.
Thank you, Lee. Thanks for that. So as Lee said, my name's Natalie. I'm an honours student with my thesis due very shortly. And I'm looking at the presence of microplastics on freshwater aquatic plants, as well as sort of a development of a standard method. So on to the next screen. So obviously plastic pollution is a global issue. Lots of people have heard, it's all in, everyone knows that the plastic's a problem. And this picture on the right was taken just down at Indy Park. So it's in the area, a lot of microplastics. So microplastics, or plastics rather, can be micro, macro, and nanoplastics. So I focus on microplastics, which also includes nanoplastics. I don't differentiate them in my research at the moment. So I just refer to them as microplastics. And they're plastics that are smaller than 5 millimeters in size. And with nanoplastics, are generally defined as being up to 100 nanometers. So a lot of those you can't see with the naked eye. So we have primary, secondary, and other microplastics. So primary are microplastics that have been designed for for use in that form. So things like resin pellets to produce plastic products, microbeads for personal care products. I mean, they've been banned here, but a lot of countries still use them. And then you have the secondary, which develop via degradation of larger plastics. So they break down into smaller and smaller pieces. And then you have other plastics. So they're things that include microplastics generated from textile fibers in your clothes, and tire wear from rubber particles from those from wear and tear. So I'm sure you've all heard about the microplastic and plastic issue. It's mainly discussed in the marine situation and the marine systems. But what I'm looking at is looking at freshwater, and prior to the 21st century, very little research on freshwater microplastics, and only 20% of the plastics in the ocean actually come from the ocean. The rest come from land and rivers, which is why I'm interested in that. So on the next slide. So as I said, microplastic research in freshwater is very limited. It's growing, but in 2018, less than 4% of all microplastic research was done in freshwater. And very little of that has been done on freshwater plants. So there has been some research into microplastics and aquatic plants in marine systems. So they've been looking at seagrasses, kelps, things like that. And they're finding that they are adhering to seagrass blades, and they found that multiple different locations and different types of seagrass. So there has been some research with freshwater aquatic plants and microplastics, but it's mainly done in laboratories. So people are getting virgin plastics, adding them to lab-grown plants, agitating them, doing different things to see if they're adhering. So they are finding that they are adhering, that it is becoming an issue. And obviously the lab work that's happening in freshwater supports what they're finding in the marine research field. So the picture on the right is a Petri dish, which is some microplastics, gives you an idea of the type of things we're looking at. For me, that's the upper limit of what I'm looking at. So the next slide. So the significance of this project is that it's been identified as an area where research is required. There hasn't been a lot of research into freshwater. So, we need to look into that. So this project, it's first of its kind in central Queensland, and even still there's very limited global research. So there's some research around identification and extraction techniques, but most of these are on soil, excuse me, water matrices, things like that. Very little is actually on plants. So we want to establish that microplastics are present on aquatic plants. We've already established it in marine systems, but we want to provide in-field evidence from freshwater systems to say, hey, this is happening here as well. It also assists in the research to develop a standard method. At the moment, there is no standard method for the identification of microplastics on aquatic plants, freshwater or marine. And it can be quite difficult because seagrasses, the techniques they use on those, they're a lot more robust and they often use scraping methods, things like that. And on a lot of freshwater plants, that's just not viable. They're a lot more delicate. So we have to look at other options. So the next slide. So the aims of the project, as I said, is to identify the presence of particles that are likely to be microplastics on the in-field collected freshwater aquatic plants. And we also want to find a cost-effective and reliable way to identify them. There are some substances that are great. However, they're just not cost-effective. You're not going to be able to use them in your average lab. So on to the next. So how we did our project was we took quantitative sample methods. There was a small pilot study, which we refer to as phase 1. And so we looked at potential effective methods and trialed a few things. So in relation to the freshwater plants, we decided to try two, one floating and one floating attached. And we used 10 grams of plant material per sample. So once we'd done phase 1, and we then looked at phase 2 to further develop the method based on the findings. So the next. So in relation to analysis, we had to digest the plant material. And that is quite difficult when you're looking at chemicals that you want to digest fairly solid plant material structure without digesting and damaging the plastics, especially being that they're so small. Oh, excuse me. So once we digested them, we filter them onto glass filter papers and we treated them with Nile red. So we are aware that Nile red has its limitations. It can stain lignin and chitin, that's part of plant cells, also some algae. So we had to look at ways around this, but still using Nile red. And we also acknowledge that microplastics generally tend to have a stronger fluorescence than some of the other items. And you can see there on the right, the picture, that's a strand of, I'm assuming, well, one would say would be plastic. You can see it's very specific, very bright fluorescing. And a lot of the plant material around it is the dark cells or the red. So once we'd stained it, we use a fluorescence microscope, and that's the picture on the right is from a fluorescence microscope. So we use that to identify potential particles. So we then used manual identification as well as software programming, software automation image J. So as you can imagine, counting particles when you're taking lots and lots of samples is very time consuming. So in phase 1, we did both and just to see if we could automate the process to make it a little bit faster. So part of my project, well, once my thesis is done, actually, I'm also going to be conducting pyrolysis and using a microscope FTIR to then look at the type of plastic so we can see what polymers are present. So on the next slide, so these are the phase 1 results. So as I said, we looked at two plants. One was Azolla, which is a floating plant, and the floating attached was Ceratophyllum. We had problems with the Azolla. We found that there is an algae that basically lives within it, and it had a lot of issues when we came to digesting it. So we decided not to continue on to phase 2, but phase 1 for the Ceratophyllum. You can see down the bottom on that graph, you've got the 3 blue bars. That's treating the plants with just hydrogen peroxide, which is commonly used for this type of thing. But what we also did, which some people are doing now, is treating with hydrogen peroxide, but also using a Fenton's reagent to have that additional reaction. And you can see from the results that in each section, the orange bars is when we use both, and it significantly increased the amount of fluorescence particles we could identify by breaking everything down. So slide is phase 2. Okay, so we only used that one species. As I said, we just stuck with Ceratophyllum. We used the same location, which is Moores Creek, obviously part of the Fitzroy Basin, and we chose a section of Moores Creek that had a lot of macro plastics. And in phase 2, we looked at the effect of different temperatures on the plants and different digestion times. So we used the same sample analysis technique to phase 1, and we used image J to identify the particles. So in phase 2, we used N equals 60. So I had approximately 84 samples, including my blanks. And for each one of those samples, I looked at 60 different locations on the filter paper and took photos of that. So you can imagine that's quite a lot of time, but we really wanted to get a good idea of what was going on. So just to give you an idea with the size of microplastics that we're looking at, both of these pictures are from the research that I've done, and both of them are 2.15 mils across. Okay, so you can see on the left picture, that looks like a really big chunk, but it's actually less than a mil. And that's one of the bigger sizes that we saw on our samples. So you can see on the right, generally a lot more smaller particles. So it is really difficult when you're dealing with very, very small particles. So the next slide. So what we found in phase 2, we used 2 options. We digested samples for either 90 minutes or until there was no reaction. And what we did before we digested, we rotted the samples. So they were in incubators, and one was at 24 degrees and the other one was at 36 degrees for 12 weeks to see if rotting them at different temperatures made a difference to them when we tried to digest them. So we found that digesting for 90 minutes was actually the better way. It digested sufficient plant material to see the microplastics. We found that if you went for a longer period, you actually got less visible fluorescence. I mean, that could be a mixture of perhaps it broke down too many plastics because of the small sizes. It could be that, you know, we broke down the organic matter so small that it was very silty, and that potentially could have covered the fluorescence on when you looked at it on the microscope and for staining. So onto the next page, we had blanks. Obviously, we had field blanks and water bath blanks. So the field blanks were obviously opened in the field. They went into the incubators as the other samples did, and then into the water baths, where the water baths were just put into the water baths. So even though we did have obviously some findings and some fluorescent within our blanks, a couple of them we actually got 0 contamination, which we're really happy with. But it's also really, really low levels when you compare what we're looking at to the other numbers. So the other numbers, you know, we're looking at on an average of, 40,000 particles projected, and an average of, 500 particles on the amount of filter paper I'm looking at versus, a maximum of 800 particles altogether, or an average, 5 to 10 particles. So it's a huge difference. So the next slide. So what we found was, as I said, we wanted to determine the presence or absence of microplastics on these freshwater aquatic plants. We wanted to determine effective treatments to identify them. And we also wanted to start a point for further research and development methods to be used in field. As I said, a lot of the research is done in labs, but we want to see what the effect is, what's happening out in our local rivers. We also want to identify types of microplastics. And if we can do that, then potentially we can look at the sources. Where are they coming from? So we've identified that, yes, there are particles on there that appear to be microplastics. We've identified that some methods are better than others and rotting times are better than others. And hopefully when we do the pyrolysis and the microscope FTIR, we can then look at these are the polymers that are present. So the next slide, where to from now? So my plan is the map on the right is a picture of the catchment rivers and drainage basins into the Great Barrier Reef. So what I would like to do is sample one, preferably 2 rivers in each catchment basin, and all rivers are to be perennial. And what we're doing is we want to look at pristine or wild rivers. So we want to look at rivers that have, no water treatment plants on them that we know release microplastics, no big known source contaminants, and see what's there. Okay. So I would just like to thank the Fitzroy Partnership for the Health for the Heart of the Basin Scholarship, providing me some financial support on this project, and CQ University for a Science Honours Stipend that I received. My references are on the next page, but are there any questions? No? So if there's no questions, there's nothing in the chat. You can contact me at any point with any questions. I'm sure that. Yep, there you are.
Muted the room. We're all yelling at you.
Yeah, I know. I didn't hear a thing. I was like, okay, no questions. Excellent, done.
Thank you so much, Al. It's an amazing topic, and just so at the forefront of research that isn't happening yet. So is there any questions? We are a little bit ahead of time, so we're happy to answer a few things if anyone had any questions. Tim? About to say
I was told to be quiet before that. Oh, look, just really interesting research there, and very worthwhile project, Natalie. I suppose is there an aspect to sort of see how these microplastics and nanoplastics also potentially work their way through the food chain?
Yes.
From aquatic life into whether they're in terms of their uptake, in terms of some other aquatic species along the food chain. Would that potentially be sort of part of that next steps?
I think it would be whether it's me or somebody else, because there's so much to look at here. I think what the main point for me is showing that, one, they're there, and looking at the size of them. So, you're looking at the size of them, or your zooplankton and small species can pick them up. And like you said, they go through the food chain. So absolutely, that is something that it's bound to be. And it's going to be interesting, the ecological effect of these starting from such a low level. So yeah, absolutely. There's definitely lots of things to look at as to, once we can show that they're there, what effect are they having on so many aspects, downstream, food chain, everything.
Tracy. Yeah.
Just a thought, Natalie. Your next phase is going to be looking at like collecting samples from, as you said, like pristine rivers. Is that going to be a challenge, finding river sources where there's perhaps no microplastics pollution, or do you find or do you think that may not be too much of a problem?
Honestly, I suspect that any river I sample, I'd be very surprised if I don't find anything, even in some of these pristine rivers. I do travel a bit with my work, and I'm lucky that I get to go some, places that are, considered, environmentally significant and, fairly protected. So I'm planning to sample from places like that. I really, really, really hope that I don't find microplastics there. But that's going to be the question. How embedded are they? Is it just from sources that we know of, or are they there? Are they from people swimming? Are they from in the air? How, are they getting in there? And if they are, then we're going to have to look at how.
Any other questions? Anything from online?
Just a reminder, if there is anything to put it in the.
Yeah, if you do have questions when you're online, there's quite a few people online. If you want to, if you have a question, put in the comments. We can't guarantee we'll ask it. No, that's not true. Thank you, Natalie. I have some questions too, but I'll probably get in touch with you at a different time. What's next? So you're working, obviously.
Yeah, I'm juggling some
Yeah.
research work with microplastic extractions from things like your Dream Buddies through the uni, and then I also work for a company that looks after water as well. So once I finish my thesis for my honours project, I still have more research to do, even though it's because I finished my thesis. I'm arranging with other universities that have some of the equipment that we just don't have access to through CQU, so I can really analyse. And then honestly, I'm going to take a couple of months off and think about the PhD and how I'm going to do it. What exactly am I going to look at? It's one of those topics that, there's so many things we can look at, and there's so many things that I haven't looked at yet that I think, oh, next time, because we had to, obviously it's an honours, we only had limited ability to do it. But because the science is so new and a lot of times we don't have it, it's a really interesting field to be in, and everywhere you turn there's something that you can look at and think, oh, I'll try this, or we could try that. So yeah, it's one of those topics that you could go on forever.
So I've got, one question from online from Nathan at FBA. How did you determine that all the fluorescence is plastic?
Well, we haven't determined that yet. Obviously, we've used the likelihood. We actually, when we did our counts for our microplastics, I forgot to say this, sorry, that we limited it to, we only counted fluorescents that were 500 microns or more in area. So because we thought the plan would be, well, let's underestimate rather than count every single glowing particle and assuming it's plastics. So we've picked up the particles that are most likely to be plastics. And I've looked at about 12,000 photos, and visually there is a lot of difference between something that looks like a plastic and looks like a cell. But how we're going to prove they're plastics, that's why we're looking at the microscope FTIR and pyrolysis. So we can go, yep, okay, those fluorescing things were there. This is what polymers we found. It won't give us abundance, but it will confirm that, these samples are some of them were plastics. And it's a start. It's very, very hard. As I said, science isn't there, especially on this, when you're looking at such small particles. So we're just using what we can and doing what we can, and hopefully new techniques will come and, the science will catch up and we'll be able to get even better handle on some of these, micro and nanoplastics.
Wonderful. Thanks, Natalie. And congratulations. Good luck with your thesis and putting that final thesis in. It's no easy task. I know that from people talking about how difficult it can be. So good luck, Natalie.
Thank you very much. And thank you very much for the scholarship and letting me present today.
You're welcome. Thanks.
Thanks.
While we're on the Heart of the Basin scholarships, if I can just welcome Katie back up here to actually present our next scholarship recipient, which we recently did announce privately, I guess, but we haven't publicly announced it.
Yeah. So congratulations to Rachel Bryan. And would you like to tell everyone what you're going to look at as part of your research?
Yes, certainly. I'm investigating groundwater input to freshwater in the Boeing River. So that's above Wonga Dam and the Boeing River catchment, and also I'm looking at it also in a couple of major tributaries. So in Fitzroy Creek, a couple of creeks there, and in Riddler Creek. In addition to groundwater going to surface water, I'm also investigating at the same time looking at the water quality, both the groundwater and the freshwater there, and also trying to identify whether or not there's any groundwater uptake through riparian trees by sampling some of the twigs on some riparian species. So
excellent. We look forward to hearing about it.
Thank you. Yeah, it might turn in 12 months.
Thank
you very much.
Thanks, Rachel.
All right, it's time to look at the report card results. And some of us in the room have report cards with us. Really sorry that you don't have them out there in the world, wherever you are virtually. Please get in touch though. We'll be sending some out to partners. And if you'd like a hard copy yourself, please get in touch with us. We want to spread the love of the partnership and the report card around. If I can now welcome online as well, Associate Professor Eva Abal. We're so lucky to have Eva as our chair of the Independent Science Panel. She leads an amazing crew of scientists who are also very lucky to have engaged with us. And Eva's also the Chief Executive Officer of the International River Foundation. So she brings a wealth of knowledge, not just regionally and nationally, but internationally as well. Welcome, Eva. I can see your face there online, so I'll hand over to you, and I'll mute the room again in here and change slides for you as you go through.
Thanks, Lee. I'm assuming you could hear me, right? Okay, so thank you very much for that introduction. In fact, I'm actually very honored and privileged to chair the Independent Science Panel of the partnership. As Lee has indicated, the partnership ensures that, the ISP, or the Independent Science Panel, ensures that there is rigor behind the report card that you have in front of you. But before I go to the details and of the report card, please allow me to introduce the members of the panel. We have Dr. Roger Shaw, we have Dr. Sue Vink, Dr. John Platten, Associate Professor Helen Stratton, Dr. Barbara Robson, Associate Professor Nicole Flint, and Cassie, or Catherine Jones, who actually is our convener and coordinator, and basically keeps us on our toes all the time, right, Cassie? For the details in terms of expertise of the members, please feel free to go to the website. You will also see these photos there on the website. So thanks for that. Before I go ahead with the results, let me share with you what the panel does when we have our meetings. Well, the panel provides independent, comprehensive, unbiased scientific advice to the Fitzroy Partnership for River Health, and also oversees the monitoring and reporting activities to ensure that they are scientifically robust, practical, and meets contemporary standards. The meetings are actually exciting too, because the panel provides any innovative trends in monitoring, which we share with Lee and her team, as well as with each other. So I think I'd like to, on behalf of the panel, endorse that the data you are seeing in the report card today has been rigorously generated, and basically has gone through an independent review and scrutiny. I would like to acknowledge, though, the partners and the third-party agencies who have provided the data for this year's report card. So with this, I think this is where we get a drum roll, right, Lee? So the drum roll for the results of the report card will be on the next slide. And basically this year, well, the report card shows that the overall catchment has a grade of C, which is fair, similar to the one last year. Just quickly, I'd like to remind you of what the Ecosystem Health Index measures. So can I go to the next slide, please, Lee? So the index measures several parameters. First, it does physical chemical parameters. It measures nutrients. It measures toxicants, mainly metals. And the reason why we do this, because metals can potentially be toxic in very high concentrations. The ecology component of the report card consists of fish and macroinvertebrates, and we'll talk about ecological data later on. And we also have, for the estuary zone, we add barramundi recruitment. So these different parameters comprise the report card. And then the way we then look at where they are in terms of rating is based on how far they are from the water quality objective. Water quality objectives are numbers or metrics that have been set for particular waterways, in this case, the river, that then allows us to ground the condition of the rivers back to the values that the community actually place on this particular waterway or river. Most of the other report cards in the region, in Southeast Queensland, actually use the same methodology. So if we go to the map, which is on the next slide, you will see the overall grades for each of the 11 catchments and the estuary zone. As Katie has indicated, we have actually added, reintroduced the estuary zone, which we will talk about more later on. So most catchments receive a fair or a C grade this year, except for the Upper Dawson, the Nagoa, and the estuary, which scored slightly higher with a grade of good, or with a grade of good, yeah, or B. So, but you might ask, why are there no A's? You know, can't waterways have an A? What does this mean? This indicates that one of the categories scored lies outside the guideline values or the water quality objectives set for that, or it actually is above the worst case scenario. So let's have a look at it more in detail. So let's go to the next map, Lee. This is the map from the report card, and it shows the individual category scores for each of these subcatchments. Apologies for that, it's kind of busy, but what you will see is for each catchment, for each grade, there's the breakdown. So we need to remember that this is for the 2021 water year, which was quite a dry year and below average rainfall. So what does that mean? So when there's not much rainfall, there's very little substances or pollutants flowing from the catchment, but at the same time, some of our waterways that need water actually will have very low water quantity, right? So this is very similar to the year before, which was actually drier than average. Now, when you look at the different categories, you will notice that there is no biological data for 5 of the 11 catchments. So the remaining 6 catchments were all low scores for this category. This is specifically for the macroinvertebrates and the water bugs, or the water bugs. So these water bugs can indicate poor water quality, but in this case, it was due to the lack of water, just because it was a pretty dry year. So many sampling sites were dry, and some of the remnant pools did not flow at all. So hence, the ecological category in most of the catchments were rating quite low. Bear in mind, again, I would like to note that a few of these catchments actually did not have biological or ecological data. So let's have a look at Nagoa, for example. So if you could probably point that out, Lee. The physical chemical parameters for Nagoa was graded good. The nutrients and toxicants were all within the guideline values. And as such, they were graded excellent with an A. And however, Nagoa had no biological data. So if you look at the confidence icon, which is that series of dots, it's only indicating 2 dots. This means that from the ISP perspective, that ISP was not very confident in the actual grade, because it actually did not have biological data for Nagoa. So there was not enough data collected, or not enough sites to provide a really good indication of the overall health, basically, which is indicated in that confidence rating. So the message here is that this is a wonderful tool to provide a snapshot of the health of the basin, but please go into detail behind the scenes to really understand what's happening. Later on, I will hand over to Cassie, and Cassie will provide you with a tour of the website shortly, so that you could go down to the detail of each grade. So if we go just briefly to the next one, Lee, before I go back to key messages, can you click on the next one, which is about the inshore marine conditions that was reintroduced this year. So we brought back inshore marine monitoring this year. And as you will see a snapshot of the inshore marine data in the lower GBR. So the marine grade is calculated from 3 different scores, quite different to the ones in the catchment. So we have coral, seagrass, and water quality. And coral is further broken down into sub-indicators such as proportion of macroalgal cover, So for those who are familiar with what we call as the phase condition, if corals are replaced by algae or seaweed, then it doesn't really indicate a good ecosystem. So it also includes the rate of coral cover change and the coral community composition, coral cover, and especially the juvenile coral density. Now, this indicator received a D for this area. Seagrass is also broken down into two sub-indicators, seagrass abundance and seagrass resilience. And similar to coral, seagrass was rated a D this year. Water quality, though, is rated a B. And as you'll see, it's got three sub-indicators: water clarity, or which is the opposite of turbidity, productivity, and particulate nutrients. So overall, this is in the inshore marine, you have two Ds, one for seagrass, one for corals, and then you have a B, which is for water quality. So if we flick back to the key messages, if I could just summarize it, Lee. So once again, a reminder that the 2022 report card addresses the 2021 water year. So another dry year for the basin. The significant rainfall we have had over the past 6 months will be featured in the 2023 report card rather than this report card. So the catchment grades this year can be attributed in part to rainfall, which as we said, below average. However, we also look at catchment condition and rainfall levels and intensity do impact water quality. So while erosion and runoff can negatively impact water quality, especially in flooding years, in times of below average rainfall, a lack of flushing can keep pollutants in stream, also causing water quality issues. So they're kept on the stream rather than being flushed down to the other parts of the basin. So the overall trend is that, once again, before I give a very quick snapshot of the trend and the overall trend, is that the data set for the Fitzroy Basin is quite strong. There are a number of data sets, data points, so many data points provided by our partners and third-party agencies. But we do continue to have gaps, and especially in the ecological and the biological data. And we would like to see if we could address this in future report cards. Over the past 10 years, the aquatic ecosystem health of the basin has remained fair to good, and this can be seen in the trends graph over 10 years, and Cassie will show that as well on the website. It seems that waterways scored slightly higher during years between 2013 to 2016 and have decreased slightly back to earlier levels. So the reporting incorporates data for both normal weather events as well as those abnormal or high weather events, or including major floods and droughts that have occurred in the basin over preceding years. So the report card actually synthesizes these different events. It is great to see that the system is resilient and that there has not been a decline over the past 10 years. This indicates that management of waterways has been stable. So resilience is really a concept, a way forward when we look at waterways and rivers and our basins, because basically the vision of resilience are rivers for future generations. And that's why talking about, using the report card, interpreting the results in the context of resilience is really an exciting process with the team and of course with the ISP. So I would like to commend all the partners, once again, the independent science panel, and especially the team of the Fitzroy Partnership for River Health, Lee, especially your team, Braden, Nicole, Eva, for your work in bringing all of these information together. So much information, condensed, synthesized, of course, with the coordination of Cassie into what we see as a very simple report card. I would like to share what I said in terms of where the report card sits in terms of science communication. The report card is a very strong instrument for science communication. It sits at the top of what we call as a wedding cake structure, where you have the bottom of the wedding cake, which is the widest, actually has the most data sets. And then it's synthesized to the next level, and the highest level is the report card. And hence the report card has the rigor in that it's being supported by several layers of data and synthesis. Sometimes we ask ourselves, well, is this too much synthesis? And are we actually simplifying everything? Well, the challenge we have now is that we need to simplify information when we provide it. And the report card that you have in front of you is the best way of actually reflecting the ecosystem health of the basin. So once again, my commendation to Lee and her team. And it now gives me great pleasure to hand over to Cassie, who will then run through with you in more detail the data and the information behind the report card. So over to you, Cassie, for the interactive web page. Thank you.
Thank you, Eva. I love the wedding cake idea. Yeah, very
Yes.
Hi everyone, so I'm Cassie. As Eva has mentioned, I'm the science leader for the Independent Science Panel for the Fitzroy Partnership for River Health. So I'm going to give a tour of the website. So I will start by, this will take us there. So if you go to the riverhealth.org.au website and go to this tab at the top, which is report cards, the hard copy report card that you've been looking at today or that will be sent out in the mail is the Ecosystem Health Index. So we click on that one. And that's the result for this year. That's the grade for the 2022 report card or the 2020-21 water year. So we have the, as Eva has announced, again, it's a C. If you hover your mouse over that, you'll be able to get the score, which was a 61. The grade is a C. This year, the confidence was 3 out of 5 overall. If you were interested in any previous years, you can select them from the drop-down menu there. But we'll go into this year's data, this year's grades, I should say. So having a look at, there we have the 11 freshwater catchments and the estuary. And if you hover over any one of them, you get a little pop-up there with the score, the grade, and the confidence in the grade. And then underneath, you've got those 4 category icons that are color-coded. So those match the hard copy there. And then, so you can hover over any one of these to get that information. I'll click down and you can click down to any catchment that you're interested in. So I'll just use the Upper Dawson as an example. Clicking onto that one. And here you can see the catchment that you're talking about has been highlighted. It's highlighted in the colour of the grade of the year. So this year, the Upper Dawson got a B, so you can see it's in that green colour. And then the grades on the right-hand side there, they directly relate to those icons. So the Upper Dawson had a B for physical chemical indicators, it had a B for nutrients, B for toxicants, and in this year, none of the data providers from the Upper Dawson have sampled for macroinvertebrates this year. It's not part of their monitoring program. So as Eva said, we are assessing the third-party data. So there was no ecology data available to assess for that particular catchment. If you click down into each of these tabs, you then understand what makes up each of those categories. So for the physical chemical category, that's made up of electrical conductivity, pH, sulfate, and turbidity. The nutrients is total nitrogen, oxidized nitrogen, total phosphorus, and the filterable reactive phosphorus. Toxicants, they are in the toxicant category, up to 17 different metals are assessed. And as you can see, not all data providers will monitor all 17 metals, so it will be dependent on which data is available for each catchment. The ecology data, as we mentioned, there wasn't any for this particular year for this particular catchment. But if they were, then of course the grades would appear here. So it comes up as an N for no data if there was no data. And in that case, the grade for a catchment is then it'll be out of 3 categories instead of the 4 categories. So you can download these numerical scores. So any of these numbers that pop up when you hover over the website, if you want to see them in a nice summarized form, you can click on the download data here and it will download whichever catchment you're interested in. So I'll let you do that in your own time. Just going to a catchment that did have ecology data this year. So if we chose Mackenzie, click on that one. So you can see that it's turned this orange color, which represents a C grade. And it has data for all 4 categories, so all the parameters within those categories. And it actually has 5 out of 5 confidence. So that means that the science panel was confident in the grade for that catchment. That's because it has sufficient data from a sufficient number of sites under both high and low flow river conditions. So yeah, so each of the categories is the same. That we graded individual grades, and then you can download the data if you'd like to see the actual score. So when you hover over something, it also has a score that corresponds to that grade using that grading ruler that's in your in the hard copy report card. The Mackenzie is one of those catchments that has quite a significant amount of data available. So it also, the monitoring programs of the people that sample out there include all the 17 metals that we are able to assess using the report card assessment methods. And so as you can see, these are the grades for the 17 different metals that can be assessed. It's actually only the grade of the lowest scoring metal that is reported. This is a conservative, cautious approach just to make sure that there's no effect of averaging. So if you could see here, there are many metals that are receiving an A and B grade, but the report card grade for the Mackenzie for the metal score is actually directly related to this lowest scoring metal. So I'll just go a little bit back up there. If you see the overall score, apologies if this is a bit too much up and down scrolling. But so the score directly relates to the copper. Now, if you wanted to know exactly how those number values are coming about, I'd recommend going to this resources section of the website, and we have the grading explained. The grading explained is broken up into the various types of reports that are provided. So we have the freshwater and estuary, the crop use, the stock use, so they'll be discussed a bit later today, but this is the freshwater and estuary grading explained. And again, you would read through that. This year, a lot more information has been summarised in the hard copy report card to get a better understanding of what goes into those grades and how the numbers convert to letters. Of course, there's always more detail and there's a lot of math that goes behind it. So I would just draw your attention to this part. Apologies for the scrolling there. So perhaps this figure is the extra detail that's maybe not in the hard copy report card, and something that's useful to go to the website to investigate is just understanding those benchmark and worst case scenario values and how every single data point that is received is assessed with being how close it is to the benchmark, and those benchmarks will be guideline values, water quality objectives, to a worst case scenario that's been defined in the development of the report card that will relate to an impaired condition. So each data point is compared to both of those values. If all of the results were above that benchmark or at the benchmark or better than the benchmark, that's how you end up with an A. If all of the results are worse than that worst case scenario, that's how you get the E. And then the ones in between are scored using this mathematical equation. So I just let you know where that is on the website for those of you that like doing a bit of maths. And yeah, I'll now just navigate back to, there was one last thing I was going to mention about the report card on this interface here that you can do that's quite interesting is you can compare different years. So using the drop down menu there, you can compare the grades over time. So over the 11 years of report cards, if you just hide that to make it clearer. So that's the overall grade. So that's what the Fitzroy Basin report card grade has been over those 11 years from 2010 to this year. Well, taking into account that it's the water year. And so you can do that for the overall grade. You can then drop down one level lower. You could do just for one catchment. So back to that. Sorry, navigating here with the mouse. So then that's just the Upper Dawson grades over time. So that was the catchment that we looked at before in detail. So that then you could say, well, what's the Upper Dawson's physical chemical category been over time? Like that. And there you go. So that's how physical chemical category for the Upper Dawson, which is part of the basin, has been graded. And then again, of course, you go a step lower. So how has the pH gone over time? And again, so it's just, now I can use the wedding cake analogy, which I'm just about. We can go from the pH up to the physical chemical category, up to a catchment level, and then up to the report card level. So I think that's all that I need to tell you about. I know it's a bit awkward using a website and interacting like that. So I think it's best if everyone just visits the website. That's theriverhealth.org.au. And I will hand back over to Braden now. So yes, Braden would like to come up.
Thank you, Cass. My name is Braden. I am one of two science project officers here at Fitzroy Partnership for River Health, and I'm going to go through the drinking water report. So what is the drinking water report? We've discussed the Ecosystem Health Index, but we also have other reports such as the Drinking Water Report and the Ag Use Report. The Drinking Water Report is an index on the health and aesthetic of drinking water within the Fitzroy Basin. This Drinking Water Report draws on information provided by the Rockhampton Regional Council and the Central Highlands Regional Council. Just like the EHI report, drinking water results need to be governed against a set of guidelines or standards to provide an authoritative reference on what defines safe, good quality water. Unlike the Ecosystem Health Index water results, drinking water is defined as water that is safe for human consumption. This drinking water report is governed by the Australian Drinking Water Guidelines. So some more about the report here. Drinking water reports have been prepared by applying the Australian Drinking Water Guidelines to the data. These results are then averaged and graded using the drinking water method, which can be accessed on the drinking water page on the website. Both aesthetic and health guidelines are used with results for treated and raw water. Raw water comes from creeks, rivers, dams, and underground bores. This water has not been treated for use as drinkable water or any other uses. Councils and other suppliers of water to townships process this raw water to make it more drinkable, potable, or useful by purifying, clarifying, softening, or deodorizing it. Here's some history. So the Drinking Water Report began in 2014 with the 2010-2011 Drinking Water Report. We are now up to the 11th Drinking Water Report, and our latest results are for the 2020-2021 reporting period. Please note that these results are for over 1 year ago. These results are always very similar, and this is great because this highlights that we have very safe drinking water here in the Fitzroy Basin. Consistent drinking water results are expected as a treatment process involved, which is designed to develop one product, safe and delicious drinking water. Drinking water results for the 2020-2021 period were mostly good, with some exceedances. The Australian Drinking Water Guidelines recognise that occasionally there may be a health or aesthetic-related test result that fall outside the guidelines, and that these results are not considered a risk to human health. The guidelines do not require a 100% result for all parameters in all cases. So what are health versus aesthetic guidelines? A health-related guideline value is the concentration or measure of a water quality characteristic that, based on present knowledge, does not result in any significant risk to health or to the health of the consumer over a lifetime of consumption. An aesthetic guideline value, which is the concentration or measure of a water quality characteristic that is associated with acceptability of water to the consumer, for example, appearance, taste, and odor. Now, if you want to learn any more, we can go back to the website and you can navigate to report cards here, select drinking water, and what you'll find is at the top we have a summary of the water results. You can see there's a little bit of a summary written here, some information regarding, you can click here, the link here, and find the drinking water guidelines and our drinking water method, which was developed by our science panel. You'll see here, similarly to the Ecosystem Health Index, you can select different areas throughout the Fitzroy Basin. And the two major zones, obviously, are the Central Highlands Regional Council and the Rocky Regional Council. You can select the A's here on the map, or you can select individual town sites or treatment plants and delve deeper if you like to see health and aesthetic guidelines and results. You can also find exceedances here. And if you want to know even more information, you can look down here and select the FAQs page, which will take you to another page with more frequently asked questions revolving around the drinking water. So that is all. I'll now hand you over to Eva to talk about our agricultural reporting. Thank you.
Thank you, Brayden. For those of you who don't know me yet, I'm Eva Dukov, and I've been working with the partnership since December last year as the Science Project Officer. So now that we've heard about the drinking water report, I'd like to talk a little bit about the agriculture report that we are also doing. So some of you may be wondering what the agriculture report is. The agriculture report produced by the partnership is a guide to indicate the water suitability for agricultural users. Similar to our EHI report card, an annual report is published on our website since 2013 to present the results. To produce this report, unprocessed raw surface water provided by third parties is rated for its suitability for agricultural users, and it's divided into stock and crop use. This data is compared to the ANZAC guidelines, which have been developed for stock and crop use for the Australian conditions and are therefore more locally relevant than other international guidelines. To determine resulting grades, thresholds are applied to selected indicators, which you can see on the next slide. So this is a brief overview of the methods behind the ag report. The first step of the framework is receiving data about water quality covering the whole Fitzroy Basin. Those data points are not specifically collected for the suitability of agricultural reporting and therefore may present spatial gaps. Once the data points are prepared, thresholds based on the ANZAC guidelines are applied to selected indicators. Based on those thresholds, scores and respective grades are created for each indicator at a specific site. Within each catchment, all the site scores are then averaged to create a catchment score for each indicator. To ease the comprehension of the water condition for agriculture use even more, an overall catchment score is created by combining the different indicators. As I mentioned briefly beforehand, there's two different types of agricultural reporting, since the water requirements for plants and animals are quite different. As part of the stock water use, there are 20 different indicators focusing on only chemical characteristics with the potential to affect animal health. There are other categories available in the ANZAC guidelines to report on suitability for stock water, but there's not enough data provided to cover these. Most of these indicators have the same threshold for different types of stock, except copper and EC, which are tested against the different stock types that you can see on the slide. Crop water use is measured similar to the stock water use, but has more indicators with the potential to affect soil behavior of plant growth. To adjust the method to the local environmental conditions of the Fitzroy Basin, commonly grown crops and predominant soil types are identified. Some of the indicators have different thresholds depending on those previously identified conditions. What both methods have in common is that if the indicator falls below a certain trigger value, it should be a minimal risk to the animal health or no impact on crop production. So why is it important to do an agricultural report? In the Fitzroy Basin, the agriculture industry is the biggest water user and is therefore heavily relying on water that can be consumed by stock or used for irrigation. Simplifying information from a complex environmental system is therefore crucial for the industry to create awareness and to help improve management decisions. The agricultural report is a useful tool to communicate findings and to help educate the community and relevant stakeholders. So now that you know more about the background of this agricultural report, I would also like to navigate you through our website. Similar to the EHI and the drinking water report, you can find the agriculture on the report cards. And there's a short summary also talking about this year's break. And there's a map coming up with all the different catchments for stock use, which is selected here. And you can also choose the crop use, and then it'll show you the grades for catchments. If you like, you can even drill down deeper and click on one of the subcatchments, and then it'll give you the grades for the specific indicators. There are some exceedances, as you can see in this list down here, although the majority of the data points fell within acceptable limits. Please remember that the grades are averaged to the whole catchment, which is a big area. So if you're interested in your specific property, on-farming testing should be considered to determine specific water quality and the likely impact on production. And then further down here, there's more methods, just like for the other report cards, that you can have a look at. You can also go onto the FAQ website where you can see the formulas behind the agricultural reporting. So yeah, that's it for me. And I would like to now hand over to Katie, who will be running our Q&A session.
Thanks, Eva. So that's all of the presentations. Any questions? Did you have any online, Nicole? Yes. Any from the room? No questions. Someone must have a question. Yes, Rachel.
With the seagrass, what area do you cover? Is it just sort of the mouth of the Fitzroy to Port Allen?
I might do that one.
Right. So, yeah, we can have a look at the map, I think, and we'll look at that. So,
Obviously, the Gladstone areas we don't use the Gladstone data, but shoalwater and yeah, that's right. Anything above that.
So here is our marine page, and from memory, we've got the Fitzroy River mouth. I think it's around Great Keppel Island as well. Cass, do you have any others by memory?
Yeah,
Oh, Eva.
And Eva Abal, did you have any other input into the seagrass locations?
So the actual, sorry, Braden, the actual locations of seagrass monitoring, did we get that, Cass?
Oh, just the general area.
Yeah,
But it definitely doesn't include the Gladstone. So it's basically where you actually, it doesn't include the seagrass data in the Gladstone area.
There's more information on the Groundwater webpage too, which, but yeah, it's obviously, enough to give us a good snapshot of this region. And they do obviously the whole reef region as well. And the seagrass, is still suffering a little bit from warmer sea temperatures. and the recent, big events like the cyclone Marsha and stuff. It's still kind of recovering, but there's some, it looks like there's some positives because even though there's less recruitment of seeds, and remembering they're a flowering plant, they were one of the things that evolved from land to go back to water. They're the only flowering aquatic plant, which is interesting. They're not like algae, it's very different. And so important, of course, because of nursery and food habitats for dugong and so on. But yeah, just remembering that, it doesn't look great, but there is a little bit of hope as far as some of the abundance recovering a little bit in some areas. Yeah. Again, it's really detailed and patchy depending on where you look at. There's some improvement in some areas, some decrease in others, but yeah.
Other questions?
Just on the marine grades, it's interesting to see water quality sort of sitting at a B, but your coral and seagrasses are sitting at that D level. In terms of that link between some of the grades we see within the actual sort of Fitzroy rivers or the catchments of the Fitzroy rivers, have we sort of made any links between sort of, say some of the values that we get around sort of, nutrients or toxicants and sort of the impacts that we see in corals and seagrasses, and really sort of correlate sort of some of those, some of those sort of, not as good results in those areas with what we've seen in the marine grades?
So you mean, Tim, specifically the freshwater sort of grades and the marine? Look, we haven't really done a lot of correlation in that sense. I mean, we know obviously that during dry years, the freshwater seems to be much more influenced by the boom and bust things. So the big heavy rainfall coming down, more sediment is never a good thing. It brings with it some toxicants. It obviously covers the coral and smothers the seagrass as well. So that can take time to recover. And that's what we're seeing, I think, in the marine zone is just that lag period. The coral again has some abundance, small recruits of coral coming back, but it's still recovering from bleaching events, over the last few years. So down south here, probably not as badly as it has up north if you're looking at the whole reef itself. And certainly the offshore coral seems to be slightly healthier than the inshore coral, which closer to the coast, I suppose, which makes sense, as you'd imagine. So, yeah, I guess this is the first time we've sort of used this data. We did used to look at marine information, so I think it's something that will develop over time. And certainly I think that plume from the big floods doesn't help. It's all related back to climate change, back to, more of what we're seeing with drier summers and wetter winters, which is crazy, and things like that. So, it's the resilience of the system, isn't it, overall, that's going to make a difference over time.
Interesting to see what we have next year when we analyse the additional data from the wet year.
Yeah, and obviously the water quality, as you said, is good in that inshore zone, and it has been good because there's been less rain, but it's taking the aquatic biota a little bit longer to recover. So even though the water quality is good, hopefully next time we measure, that will indicate that then that's had some time.
There's that lag aspect.
Yeah, that's right, that lag period. Yeah.
So I've got a question from the floor. Francis, the report card says there's improvements coming next year. Can you talk us through what they may be?
Yeah, sure. Thanks for the question, Francis. So the partnership has been working on, I guess, how we can get better spatial coverage for a number of years now. So we're pretty excited that we do have a new basin-wide monitoring program. So we did collect data over, I guess, the water year for next year. So we'll be able to run that through the report card, and that'll fill a number of gaps, but we're particularly excited about the macroinvertebrate data that we'll get from that program.
No other burning questions? If you think of things, remember that Nicole's got a couple, but if you do think of things later on, please let us know. We're always available to answer things on the phone or email. We're half an hour ahead of time, or 20 minutes ahead of time, which isn't a bad thing, I don't think. We'll probably hang around in here and have a bit more of a chat. But please let us know if you're online, if you've got any questions or any follow-ups that you like.
Just got one from Councillor Kirkland, quite specific about the Fitzroy crop use report. But yeah, we might potentially get back to her on that one.
Yeah, thanks very much. I know Councillor Donna Kirkland, one of our partners, is online today. I'm not feeling 100%. So we're missing you, Donna, but yeah, we'll see you soon. Well, thanks very much, everyone online. We'll let you head off with the rest of your day. Thanks for joining us. Please let us know if you'd like some report cards themselves, some hard copies. Don't forget to go online and drill down to that detail as we talked about the wedding cake, which I'm going to use now from now on. I love that. Yeah, I'll hand over to Katie to close.
Yeah, I just want to thank again the independent science panel, the team at the partnership, as well as all of the partners for their contributions to this report card, and remind everyone about our event at the end of August, and look forward to seeing you all there. Thank you.
Thanks, Katie.